Good morning. My name is Commander James Palma. I'm the President of the Faculty Senate, and I'm here to kick off our Packard Lecture. Warm welcome to all of the many general level, flag level officers and senior leaders of NIH and other civilian institutions. Too many to list by name, but we appreciate your time and your participation in this event. So it is my distinct honor as the USU Faculty Senate President to invite Dr. Stanley Prusner as the 2016 David Packard Lecturer. This is the most prestigious lectureship at the university and is named in honor of the former Deputy Secretary of Defense, founder of the Hewlett-Packard Company, and the second president of the Uniformed Services University. Dr. Stanley Prusner is a professor of neurology and biochemistry and the director of the Institute for Neurodegenerative Diseases at the University of California, San Francisco. While at the university, Dr. Prusner discovered an unprecedented class of pathogens that he named prions, infectious proteins that cause neurodegenerative diseases in animals and humans. Dr. Prusner's contributions to scientific research have been internationally recognized with numerous prizes, including the Richard Lounsbury Award, the Albert Lasker Award, and the Nobel Prize. Dr. Prusner received his undergraduate and medical training at the University of Pennsylvania and his postgraduate clinical training at UCSF. He is the editor of more than 11 books and more than 500 research articles, as well as 50 issued or allowed U.S. patents. He is a member of the National Academy of Sciences, the Institute of Medicine, the American Academy of Arts and Sciences, and the American Philosophical Society, and he is a foreign member of the Royal Society of London. Currently, Dr. Prusner is also the president-elect of the American Neurological Association. His research has many ties to ongoing USU exploration into TBI and other neurologic disorders. Dr. Prusner, it is our great pleasure to welcome you back to USU.
Well, I'm thrilled to be here, and it's a very special honor to give this lecture. I want to thank Chip Rice for asking me to do this and to tell you about some really very, very exciting new results in the field of chronic traumatic encephalopathy. Now, it's a little different than what happened in the Congress, and we'll go over that. But it's all about 7 words, and you'll hear more about that. But I wanted to start out with these pictures. And now, you know who this is. And the reason I put this up here is that Chip is going to be retiring. It's a very sad day for me because I've gotten to know Chip, but actually I'm hoping he'll come and visit us more often. And down here is David Packard. Now, how many of you know who that is? Well, obviously that's Bill Hewlett. And I thought that David Packard was the first president, but fortunately, with these computers, I could change it to former. Now, this really is Chip Rice. But I asked Dan Pearl for a picture, and he sent me this one. And well, that's not Chip Rice. He's looking very concerned and presidential. All right, now let me start out with really the major issue that we all are interested in here. And that is, do some types of head trauma result in the production of tau proteins that turn into prions? And if that's true, then can we intervene with medicines? Can we develop prophylactic medicines? Can we develop medicines that can be used in the later stages to stop the horrors that patients experience? Now, there's a lot of controversy, and I start to believe things, and then I find out that the same people who wrote with such conviction write that maybe that's not true. But what's clear is that there are people who develop horrible psychiatric problems, and we tend to call these post-traumatic stress disorders. And then there are people who develop concussions. And we take those people, we don't call them psychiatric in terms of their problems, and psychiatrists don't look at their problems generally. But we tend to, when they're advanced, we look back on the concussion in terms of a neurologic or neurosurgical disease, and then later on, we look at these people as neurologic problems, but in between, the psychiatrist tends to see them. So these symptoms overlap. And the question is there a spectrum that goes all the way from PTSD to TBIs and CTI? And is this a straightforward neurodegenerative disease process that continues slowly on and on and on? And the answer to this is I don't know. These are wonderful problems that some of you young people need to think about. Now, I think as we get medicines, we'll also get PET probes. And that way, we'll be able to define these illnesses, I think, with much more precision. Now, CTE has been studied extensively, and one of the people who's done this for years and years and years is Dan Pearl, who's here. And Dan was one of the early people who looked at the differences in neurofibrillary tangles between people with frontotemporal dementias, which resemble CTE in many respects, and people with Alzheimer's disease. Very, very different distribution. And one's always worried about a highly prevalent disease, Alzheimer's, as being the problem on the background of trauma. The term neurodegeneration generally refers to a progressive deterioration of neuronal function, leading to impaired activity of the nervous system. So when I asked Dan, how do you define a neurodegenerative disease, he looked at me and smiled, and then he wrote something down, and I said, I don't really think that's a very good definition. And there aren't great definitions of these words that we use all the time. So I tried to write something, but I've just given you a bunch of examples. Some people would say neurodegeneration, these are cell diseases. The cells are sick. That's in contrast to larger systems, such as strokes, where a whole area of the brain doesn't function anymore, or brain tumors, in which the cells surrounding the brain tumor don't work. Those are not neurodegenerative diseases. All right, so some of what I've been talking about seems mundane, but it's, I think, really important to get these words straight. Now, I then want to introduce this word that I think all of you know who are in this audience, prion. These are alternatively folded conformations of a protein. And the prions were found in scrapie, CJD, mad cow disease. It's the first examples. And then lots and lots of data has been accumulated in many different systems. So we know about yeast prions, we know about other fungi, non-yeast, that ever have prions. We know about prions in many, many different animals. And we know about normal prions, or functional prions as some people call them, and we know about disease-causing prions. So there's mounting evidence that prions cause all these different diseases that I've listed here. Alzheimer's disease, Parkinson's, multiple system atrophy, the frontotemporal dementias, CTE, which is one of these frontotemporal dementias, ALS, and Huntington's. And the data keeps accumulating. And as I mentioned a moment ago, currently there's not a single drug that stops or even slows one neurodegenerative disease. Drugs like L-DOPA treat the symptoms in Parkinson's disease, but the symptoms continue on. Now, I started being interested in Creutzfeldt-Jakob disease and began with the studies of scrapie, of sheep and goats, that had been transmitted into mice and hamsters. And the whole goal of this work was to get these little blue spheres into a bucket with none of the junk. And I became a student of macromolecular purification just across the street at NIH when I was there for 3 years and worked with Earl Statman, who was a brilliant enzymologist, bacterial biochemist. And he would say all the time to me and to everyone else, dirty preparations give dirty results, and clean preparations give clean results, or more interpretable results. And I also learned from him that when something is not well understood, you need to keep challenging the system. You need to keep trying to overturn what you think. And that is the most valuable thing you can do, because if you come out with a view that is not well grounded, it takes years and years and years to undo the damage that you have done. So I've tried hard over the years to make sure that when we publish, it's as correct as possible, leaving no stones unturned. Now, the key advance was with moving from mice to hamsters. So we were able to look at tighter preparations in 70 days instead of a year, and we were able to use 4 hamsters instead of 60 mice. And so the work went unbelievably fast by comparison. Now, as we purified our samples, this is a relatively simple purification with detergent extraction, nuclease digestion, and then the key was limited proteolysis, because limited proteolysis allowed us to purify the sample, get rid of about 95% of the contaminants, and then do the last cleanup on sucrose gradients. What we found was a surprise, because all along the way, we were sure that eventually something that replicates would be based on nucleic acid, and we would find this dependence of nucleic acid, and that this would all be removed, meaning the cover and the nucleic acids would be revealed. But that turned out not to be true, that only procedures that destroyed proteins would inactivate the Scrapie agent. Now, a lot of this work actually happened at the NIH under Carlton Gajicek and Joe Gibbs. And they were really the leaders of this field for several decades. And they were the first ones to transmit Kuru, the first ones to transmit Creutzfeldt-Jakob disease to chimpanzees. There were other people in Britain who were really the leaders in what I would call the rodent biochemistry, where what happened in the rodent field was that people were able to develop a mouse assay to take the place of a sheep assay. And then what people were able to do is to try to probe with these crude preparations the resistance to damage to nucleic acids and damage to proteins. There were some really quite fascinating studies by Tigve Alper and her colleagues suggesting that there was no nucleic acid, but people were unhappy with that idea. Now, there was a lot of unhappiness as we became more and more sure that we needed a different term instead of a virus or a slow virus or an unconventional virus. And so I suggested the term prions, and there were a lot of unhappy people. That's me with brown hair. I wasn't born an albino. And so it became clear over the years that prions are composed of a host-encoded protein that adopts an alternative conformation, which is self-propagating. That's really the difference between a protein that can shift from one conformation to another Here, once this new conformation occurs, it becomes self-propagating. And generally, prions are enriched for beta sheet, and they assemble into amyloids. Now, people obviously want to know who these guys were. I show you them. And they don't need a lot of time on the screen. This needs more time because I'm always now trying to sell books. So I just want to make sure that I mention this book because there's a lot. It's my view. Let's be clear. This is my view of how this whole area opened up. And the book begins with my current. I have one grandson, and he's age 8, and that's the same age as my grandfather, who came to the United States from Moscow in 1893. And I have to say, this is a pitch for the University of Pennsylvania because I became excited by science and by education. Before that, I was so bored in high school, I couldn't stand it. I really didn't want to go to college, but I couldn't find anything to do. And then I talk about all the excitement of doing biological research, but really I started out wanting to be a cardiac surgeon. Don't ask me why, Chip, I'm sorry. To sound negative, but I didn't like that idea, and eventually I became a physician scientist. And when we view these diseases now as this group of diseases, we have all these names, and this is what neurologists are quite good at. They're quite good at keeping all these names in their heads. But even better are these simple proteins that are the basis of many different diseases. So with mad cow disease, scrapie, chronic wasting disease, Kuru, CJD, this is the PRP protein that turns into a prion. In Alzheimer's disease, it's A, beta, and tau. I'll say more later. Then there's Parkinson's disease and Lewy body dementia and multiple system atrophy. That's alpha-synuclein, and I'll talk much more about multiple system atrophy. And then there are frontotemporal dementias, and of course CTE is one of them, but there are many others, and I'll talk about PSP, progressive supranuclear palsy, cortical basal degeneration, Pick's disease, and our gyrophilic grain disease. And you can tell that neurologists and neuropathologists, Nan, you're part of this, love these crazy words. And tau is the offending protein, but there are others that can behave in the same way with respect to some of the frontotemporal dementias, ALS and Huntington's disease. One of the really exciting discoveries that we made was when George Glenner came from the NIH on sabbatical to San Diego. He was touted to me as the only person on the West Coast who knew anything about amyloid. So one day, in 1984, 83, with Mike McKinley, a postdoctoral fellow, with a box of ice and a sample of our most purified preparations that came from hamsters infected with scrapie prions. We got on an airplane. We just walked right onto the airplane with a little box of ice, and we got on the plane. Plane landed, we walked off the plane, we went and rented a car, and we drove to UC San Diego, where George Glenner was. And when George looked at the bright field, because he had stained the preparation with a little Congo red dye, and then used the polarization microscope, and he saw this green, gold, green, or birefringent pattern, he got really excited. Now, that fit with what we had seen by electron microscopy. And the reason we went to see Glenner is because this looks so much like amyloid. But it took me months to stumble into a book on the electron microscopy of proteins and look at the amyloid chapter and say, this material all looks the same. One amyloid looks like another. So none of these are alike. One of the things that's remarkable about viruses is that they're all the same. If you've seen one poliovirus, you've seen them all. There are a few that have empty capsids, but the complete virus looks the same. That's not true of these amyloids. There's slight differences. And that opened up the idea that all of these different deposits in the brain, such as the plaques of Alzheimer's disease, the neurofibrillary tangles, the Lewy bodies, the plaques in CJD and in Kuru, might in fact hold the key. They might be the etiology. Now, tau was a special case because tau was so complicated. And when you made a list of all the diseases that tau participates in, all the way from Niemann-Pick type C disease, which is a metabolic disease, to Alzheimer's disease, to the frontotemporal dementias, you really were confused. And even rabies virus, you see tangles that are filled with tau. So that didn't help us a lot. That made us more confused. But the world of CTE was extremely important, and I'll talk about it a little bit now, as it began to bring clarity to what was going on. And of course, a lot of this came from people who had played contact sports and then committed suicide, like Junior Seau. Now, this is what I said about the Congress. So if you read up here, you say, this sounds like from Abraham Lincoln. Perhaps no one will remember the setting. A hearing room in the House Energy and Commerce Committee. I have to be able to read this, sorry. Or the person who asked the question, a member of the House of Representatives from Illinois. But 7 words spoken in the Rayburn House building in Washington on Monday could profoundly affect the country's most popular sport. So then the issue starts to move on with a roundtable discussion about concussions. And is there a link between football and degenerative brain disorders like CTE? The NFL's vice president, Jeff Miller, then is asked, is that true? And he says, and there are 7 words here. The answer to that is certainly yes. Now, that's a huge change. It's not based on science, it's based on commerce and the threat of something horrible happening to the National Football League, like its disappearance. The mothers of children, young boys, might just prohibit them all from playing football. I think we owe a huge debt of gratitude to people who have been studying this disease process, because it opens up more than just the issue of football. But here's a young man who, so I focused on him because he went to the University of Pennsylvania. And in his junior year, he was selected to the All-Ivy team. And he had played football from age 9. He'd never had a concussion. I don't believe that. I don't believe the next bullet, where in April 2010, at 21, he committed suicide. And he had never had any history of psychiatric problems. It's just instantly occurred? I don't think so. Now, what Anne McKee found in his brain was all of these collections. So this is the brain stained with anti-tau antibodies, and then you see around some of these blood vessels, you see collections of tau. And this may, in fact, come from the intravenous pressure from blows to the belly that then raise the pressure inside of the brain. We don't understand these details, and these are the areas that we're trying to work very closely with Dan Pearl to unravel. Now, this man, Michael Borich, played for 2 small college teams. And again, this is all Anne McKee's data. And at age 40, he was the defensive coordinator of the year at the University of Utah. but his behavior started to deteriorate, and he was let go, and he went to live with his father, who found him very difficult. So he was drinking, he was using drugs, and he probably died of a drug overdose. And what you see are these larger, much larger collections of tau. And when you look down here into this field of tangles, I've never seen this many tangles. The intensity is extraordinary. But tau deposits come in all kinds of forms. And we're going to discuss the 3R and the 4R isoforms in a moment. But in Alzheimer's disease, the neurons get filled with these really densely packed polymers. They're filaments of tau. In our gyrophilic grain disease, you see more little deposits, but they're widespread. In Pick's disease, there are Pick bodies that are adjacent to the nucleus within the cytoplasm. And here, in progressive supranuclear palsy, we have widespread filaments, the long axons filled with tau. And over here, corticobasal degeneration, in many respects, looks similar at the microscopic level. Now, these animal studies have helped bring a focus, but I'm going to show you, even more importantly, from my point of view, we've come even further with cell studies. And what's happened is that animal studies with mutant fragments of tau have really opened up a new thinking about the biology of tau, that this is really a prion. And some of these studies have been carried out where the animals become ill after about 200 or 250 days. Not a very satisfying model because it takes too long to get data, but that you can transmit into these animals tau deposits. And this is really the work of Michel Godert and Marcus Tolnay at Cambridge University and in Zurich, where these kinds of deposits can be found. The cell work has been primarily carried out by Mark Diamond, and then some work by Virginia Lee and her colleagues at Penn. Mark Diamond is now at the University of Texas Southwestern. And I'll have a lot more to say about that in a moment. So we can find these deposits of tau in the brain. We can find that these deposits take up a lot of space, but the animals don't die. Death in an experimental model is the very best endpoint. And that's not true with tau. It is with MSA, and I'll talk about that. So, as I mentioned already, there is this interface between psychiatry and neurology. And that's extremely important to understand and to think about, because what happens in all the frontotemporal dementias is that the patients are generally seen by psychiatrists for years. And late in the disease, as they began to develop clear neurologic deficits, then they're referred to neurologists. So it starts out with changes in personality, behavior, executive function, difficulty planning tomorrow, the next week, planning a vacation, writing a business plan, envisioning anything that takes some of these higher cortical functions, particularly the ones in the frontal lobes. And these behavioral changes can then march from lethargy to disinhibition. And they can include apathy, social withdrawal, socially inappropriate behavior. Insomnia is common. And then many of these people end up with drug addiction and alcoholism, and that's the setting in which they commit suicide. Now, I wanted to summarize the connection between PTSD and TBIs or CTE, and through these Venn diagrams, because we really don't have all of this information. And I want to make sure that everybody who walks out of here, I hope all of you walk out of here, that you understand that this is not well understood. That's really important, because this is a great area of research that needs to be focused on. So there's an interface between psychiatry and neurology that wasn't, I think, appreciated very well by a lot of people, particularly neurologists studying the frontotemporal dementias. But it's quite clear now that this is a continuum and that this interface is key. And we need to stop right here. Right here, we need to stop this process. Now, how do we do that? What happens with tau is that the protein becomes misfolded, and then these misfolded proteins self-propagate, and eventually we get these aggregates or fibrils, and they turn into tangles. This is a complicated slide, and you don't really need to remember it, but what you want to understand is that there are these different isoforms, and there are 6 isoforms, and there's alternative splicing near the N-terminus, and then these repeat regions of about 30 amino acids each are the regions that bind to microtubules. And that binding of tau to microtubules seems to accelerate the formation of microtubules, but it's not necessary. If you knock out the tau gene, the animals do fine, and they have microtubules. So there are 6 isoforms, and as I'm going to talk to you about the difference between the 4 repeats and the 3 repeat taus are going to feature in what I'm going to talk about. Now, these diseases have been classified by 3 repeat and 4 repeat isoforms, and then the mixed tauopathies. And until recently, this led absolutely nowhere. It was something to remember, but you had no idea why this was important. But this has been around from the early 1990s when the tau gene was sequenced. So we really didn't know what to do with this. But I'm going to tell you about this in great detail. With Pick's disease, you need to remember 3R. With progressive supranuclear palsy, 4R. I'll also show you cortical basal degeneration, our gyrophilic brain disease, and then I'm going to show you these mixed tauopathies, CTE, and Alzheimer's disease. Now, Mark Diamond worked for about 15 years trying to work on systems like Huntington's disease and later tau. And what he was trying to do was to develop a FRET assay so that he could understand, using that process, these diseases. And he had a really tough time until he was able to assemble a fusion protein that looked like this with about 180 amino acids, the K18 region as it's called, which contains all of the repeats, either 3 or 4. This fusion protein with a cage, this is YFP, the yellow fluorescent protein. It's a genetically engineered version of the GFP. That and this linker region, that this protein could now be expressed in cells, and if you added prions, tau prions, you could stimulate the formation of new prions because this protein was being expressed all the time, and when there were prions present, then the tau would bind one after another. It would cluster, and you get these dots. And you don't need to read this out with antibodies or any additional dyes. You can simply look at the clusters of this fusion protein, and you can measure the number, or you can measure the area. And what you see up here with 3R tau is the only one that's positive is Pick's disease. So there's this homodimer interaction that occurs. And the same thing's true for PSP and cortical basal degeneration. It's positive here, and the controls are negative. You see the background is extremely low. Now, the cells that we initially obtained from Mark Diamond we used for more than a year, and then we started to make our own cells, and we got much higher expression. And it turns out that that higher expression gives much more fluorescence. And the background, it goes up a little bit, but not much. And you see almost every cell is full of prions. And this is PSP. And the same thing's true for cortical basal degeneration and our gyrophilic grain disease. So this is providing us with a much better method of measurement. But for a long time, we couldn't get any signal with CTE or with Alzheimer's disease. Here's PICS that's positive with 3R and with 4R. It's PSP. But here we had no positives. Sometimes the background was a little higher, and here just a couple of dots, but that's not enough to believe anything. All right, so now we looked at, these were cases from Ann McKee, and here are 7 cases of CTE. And here's the positive, the PSP, and here you see the data at 4 days and 5 days. Because these are live cell assays. You can keep read. You can do kinetics. You can read them out every single day, and it's very fast. So here's PSP, but these are all negatives, and there are 4 more negatives. Now the breakthrough in all of this was this higher expression, because when we did that, then we ended up with here's an Alzheimer's specimen, a CTE specimen, and you see huge numbers of prions that we can now measure. Well, why are we doing all of this? Why we're doing this is to make drugs that can stop all this. And it doesn't take a lot of imagination to realize that if you can make a drug that prevents all this from happening in cell culture, it's likely that you can engineer this drug for people. And of course, you can do this in animals first. And that's what I've shown, where you start with hundreds of thousands of compounds. The ones that you're interested in are called hits, and then you modify these, you make leads, and with advanced leads you go into animals and then into people. So this is some of the pieces that we've been using. This instrument is fantastic. This is made by General Electric. It takes CCD pictures of the individual wells. There are 384 wells, and in about an hour it scans all of these wells, takes pictures of every single cell. There are about 2,000 of them, and then we can analyze. We don't do this. The computer analyzes the data for us. And then, of course, we optimize the hits, develop leads, and then we're getting very close now to animals, and we hope that this will go to here sooner rather than later. Now, this just gives you a little more of an overview of the process. So if you sit and you watch, not football, basketball, okay? And you now put these different symbols in for different compounds, and you start with a couple 100,000 of them. You can start screening. And eventually you can create families of similar compounds. They're shown by colors. And then you can optimize for potency, stability, blood-brain barrier penetration, and the list goes on. And ultimately, efficacy. Now, why am I just not, I'm not telling you a fairy tale. Why in the world is this going to work? Why should you believe me? I know Jonathan Woodson didn't believe me initially. He's interviewed too many people who don't know everything. And I like to tell people I know everything, but it's not true. But over a 10-year period, what we did do on a much smaller scale, we screened about 50,000 compounds. We started with 10,000. And we developed 10 drugs that actually increased the incubation period for mouse scrapie and also for chronic wasting disease. But unfortunately, these 10 drugs do not work on humanized mice. So we never could collect data that would get us an IND from the FDA. But we never wanted to do that because that was our break point. We either got it to work in humanized mice expressing human PRP or we didn't. So while we've learned a lot about this, we learned mostly that we could actually make drugs and delay a prion disease. And this got to be pretty impressive. So we would start here, or we'd start out here with a drug. So we're giving one of these drugs, and you can see that we can double the incubation time by delaying, and then it goes all the way out to here when we give the drug. But most exciting was to give the drug prophylactically 2 weeks before, and you see we quintupled the incubation time. This is the incubation time right here with no drug. All right, so now what we've done, I've told you about this prion assay. We can take the brain from a mouse expressing tau, that's mutant tau, and that's the same construct found in people with progressive supranuclear palsy. And we can precipitate the prions using phosphotungstic acid or other polyoxometylates and collect the prions, add them to the culture, and now we can 4 days later detect the number of prions. So the idea would be to suppress this, to prevent it. And what we're looking at here is a PSP patient, and at the same time, from this large group of cells, we now have cells that are also from a mouse model, where the mice get sick eventually after 300 days, after 200 days, in another mouse model. This one is from Virginia Lee and John Trojanowski, and this one from Michel Godert and Marcus Tolnay. And you see in all cases is about the same. So what we're doing in mice and what happens in humans is quite parallel. And when we now use one particular drug and we inoculate with these two different mice, with human inocula, and then this artificial clone that we made, we started out with recombinant tau, and now we give increasing amounts to the drug, you see that the prions are disappearing, and the EC50 is about the same for whether it's mice cells or human prions. And when we look at human prions and we look at the recombinant artificial prion that we made, and we look at 60 different drugs, and we're looking at their EC50, so that's the effective concentration of 50% reduction. We see that it doesn't matter. We get these points fit within this line. So we get the same data whether we have an inoculum that's from a human or the inoculum from the clone. This gives us a lot of confidence. Now, in these mice made by Michel Godert, these mice get sick and die about 200 days of age spontaneously. They're expressing a mutant, the same mutant found in PSP, P301L or P301S. And what I want to show you is that with time, the number of prions that are measured by this cell assay go up, eventually to a point that kills the mice. Males and females are the same, blue and red. And out here at 12 weeks, I want to show you, when you look by immunocytochemistry, you can't really see anything. At 28 weeks, when the mice are dead, you can see a lot of tau deposited. And that's shown here. So at 12 weeks, the hippocampus, you see nothing, and you see a few deposits of tau. This is, again, antibodies. The green is tau, and the red is GFAP. Out here at 28 weeks, there are many deposits of tau. And, excuse me, and you see that there's really nothing. Now, what I want to give you is the summary of all these compounds. So we've tested 300,000 unique compounds, and this is now for tau drug discovery. We have 155 confirmed hits. And then what we did was to break these into series, or bin them according to their structure. And here, series A, we had the highest number of compounds, 10 of them. So we're looking at 1 in 1,000 here. And now we're looking at, we have 10 out of 155. We've now made 1,650 unique analogs. And we've looked at 80 of these compounds for pharmacokinetics. So a very large number of these analogs don't work, or they're so toxic to the cells that they kill the cells. But we can't predict this completely. All right, and now we have 5 compounds that look very good on these early tests. So we have enough we can do a 24-hour test in animals. But now we're making lots of these compounds, meaning hundreds of grams, and we'll be able to test them over 200 or 300 days. Now, the last part of my talk, I want to talk about synuclein. And the reason I want to talk about synuclein is that having a second prion that is amenable to study in the same way, but and having one that actually kills the animals in a short time is huge in the development of these tau therapeutics. So we're looking at synuclein, and as I've mentioned to you already, Parkinson's, Lewy body dementia, multiple system atrophy, these diseases are all caused by accumulations of synuclein. And Parkinson's disease can be caused by mutations in the synuclein gene. This is a Lewy body. Now, Parkinson's disease is fairly common. 1.5 million people in the United States have this. I really don't know what this number is. Dan Pearl and I have talked about this multiple times now, of the number of people with MSA. So for some of the internists who are here, they remember Shy-Drager syndrome. And Milton Shy actually was here at the NIH and the head of the neurology division before he went to the University of Pennsylvania to be the chairman back in the 60s. So we don't know the frequency. Now, these patients don't live as long as Parkinson's patients. They die within 6 to 10 years. And brain extracts, now this number is 14 out of 14, I'll show you the data in a moment, kill these mice that carry a mutation in the 53rd codon. And over here, you see that people with MSA in their oligodendrocytes have these accumulations in the cytoplasm, and so people call them glial cytoplasmic inclusions, or GCIs, another term. So we've built up a study just like the one with tau, same chemical library. I'll tell you the results of that in a moment. And we end up with these small inclusions that are based upon the synuclein coupled to YFP. Now, if you look over here, MSA cases 1 and 2 and this control were published in November of 2013. And we did that because we didn't have any more data. And I thought it was important at least to tell the scientific community that this was the case. Turns out that nobody cared. And nobody has written a follow-up paper that I know about. This was published in August of 2015. So we collected 12 more cases from 3 continents, and you see they're all positive here. Of all these animals studied, there are 135 animals studied, and 134 of them are positive. Only this one, this initial case, never came down. But 7 out of 8 did. It was 1 animal. And then we developed, and this is Amanda Warman and Steve Olson who developed, and Atsushi Oyagi. We developed a cell culture assay. And this cell culture assay is positive for each one of these. And then when you look over here at all these massive numbers of controls, you see that they're all negative. Here's a positive down here. Now, this is an example of why it's so great to have a second system, because it shows the selectivity. And there have been papers published that say that tau interacts with lots of different other proteins, and some proteins mimic tau. But you see here these tau fibrils. So these are recombinant tau proteins that then form fibrils and then are added to cells, and you see the positives. And the same is true for synuclein. But it doesn't work if the expression is tau YFP in these cells. The converse for tau fibrils doesn't work with synuclein. And the converse of synuclein fibrils with synuclein, of course, it works. So this is hugely important in all of these, the tau YFP and the synuclein. These don't work with A beta and PBS. Now, we ran out of a few of the compounds, so we only screened 185,000. And our initial hit number was 802. But the confirmed hits were 26. And some of these hits were studied in Japan, because we have a collaboration with Daiichi Sankyo. And these confirmed hits, we had 18 from Japan and we had 10 from San Francisco. We had 2 that overlapped, so we ended up with 26 instead of 28. But what's amazing is that there was no overlap between the synuclein screen and the tau screen. So here we're looking at 200,000 different compounds, and none of these overlap. From this huge library, we don't find a single overlap. We'll probably find one, but this supports what I've been saying all along, that here we have these unique proteins. They're different from each other. They're clearly different proteins. They don't share amino acid sequence. They share the ability to adopt an alternative conformation and undergo self-propagation. There are a lot of people who have suggested that a single drug is going to work for all these prion diseases. They're all neurodegenerative diseases. But I have never believed that, and this data argues that that will be the outcome. But until we get drugs, we have to reserve, I think, our views a little bit. So now these 26 hits, we bin them into different series based on their structures. Here we've made 1,677 unique analogs. We've done the pharmacokinetics on 17, and we have 2 compounds for in vivo efficacy. And they look better than tau, but we'll see, because we don't know yet. So what I've told you, these are the points of intervention. We begin with DNA and the prion precursor, and then the prion is formed, and this is a second point of intervention, which is what we focus on, because we're trying to prevent new prions from being made. And as we lower the prion load, we're letting 3, which is the clearance, take over and destroy the prions. And what you want to do is to lower this formation rate below the clearance rate, and ultimately the prion infection will disappear. And this simply shows you that these people who have a mutation, this age-dependent mutant prion formation that occurs happens later in life, but it's the same process. The energy barrier, the energy minimum, is much lower for the prion to form, and so at an earlier age we end up with mutant prions. But it's not age 3 or 4 or 5, it's usually people in their 30s and 40s when this happens. Okay, this is my last slide, and I want to put this up here so that you don't get too confused. Now, what am I saying here? So when you read the literature, the current literature about prions, you see there are some people who call prions prion-like protein aggregates. Others call them transmissible proteins, templated proteins, prionoids. That might get the prize. Proteopathic seeds, misfolded proteins, self-propagating strains, protein pathogens, quasi-prions, pseudoprions, amyloid assemblies, proteinaceous nucleating particles, or protein assemblages. Now, why do they do that? What's their excuse? Their excuse is that unlike Creutzfeldt-Jakob disease, where there has been transmission of few cases, compared to the fact that we have about 1 in a million people on the planet develop CJDs sporadically, spontaneously, each year. And because there's no evidence for transmission between humans, they want to say these are not prions. And of course, that's nonsense.
I'd like to thank Mr. John Lowe, the CEO of the Jackson Foundation, for its sponsorship of the Packard Lecture. And Dr. Prusner, thank you so much for being our Packard Lecturer today. I have 2 things for you. I noticed that tie you're wearing. Is that from the Trump collection? Or so here's a Uniformed Services University.
I use dye, just like Donald.
And we have a
Isn't
bowl for your
This looks empty.
It's not. This is for your desk. Thanks again for coming.
Thank you.
Please join us for a reception out in the Hall of Flags. Thank you all for coming, and Dr. Prusner, thank you again so much for a very stimulating lecture.